So this talk is being recorded for the YouTube posting, and basically it'll be recorded for, I guess the next hour or so. Okay, great to see everybody here. I'm pleased with turnout. Um, you know, when I started talking about, or started thinking about what to talk about today, nitric oxide came to mind. It's an old friend. It's a molecule that I've been studying for at least 20 odd years. So without any further ado, let's get started. Alrighty, so nitric oxide has many faces. It shows up in the air. It shows up in things that we eat. It shows up in other places. I'm going to talk about each of these things, perhaps the Cialis and Viagra a little later in the talk after you've seen some more of the biological type chemistry. But it does show up in meats and biological systems, certainly in the atmosphere. Alrighty, so good question. What is NO? It's a small molecule. It's got one nitrogen atom and one oxygen atom. And here's the thing. Nitrogen has 5 valence electrons. Oxygen has 6. There's an odd number of electrons. There are a total of 11 valence electrons. So, and only 2 atoms. There's no way to divide these up so that you get every electron in an orbital paired with another electron. This gives nitric oxide some interesting properties. One property is that it can be detected by electron paramagnetic resonance. And in fact, in biological systems, the very first detection of NO, before we even knew what we were detecting, were by EPR. It makes a beautiful 3-line signal. And this EPR technique is extremely sensitive. You can pretty much detect maybe 1 billion spins in a sample. That sounds like a lot, but when you consider just how many atoms there happen to be in just a gram of like hydrogen, the mole, 6.02 times 10 to the 23rd, 1 billion is not a lot. Nitric oxide occurs naturally, but it requires energy to make it. It can be made from N2, dinitrogen, and oxygen in the atmosphere, but again, it requires energy. There is no danger of the nitrogen in the atmosphere ever catching fire and just burning as a fuel to make the various forms of nitrogen oxides. Takes quite a bit of energy. It can form in lightning. It can form in car energies, and there are various forms of nitrogen oxides. I've listed a few of them here. There's N2O, which is the nitrous oxide. Nitrous oxide is the laughing gas that is used as an anesthetic, perhaps in most famously, I think, in dentist offices. Yes. So nitrous oxide can be inhaled. Nitric oxide, I suppose it can be inhaled, but immediately after you'd have coughing fits because nitric oxide smells pretty much like, I don't know, concentrated acid. It burns. It burns. Nitrogen dioxide is the next one, this NO2 guy. It burns as well. Very, very unpleasant stuff. And then there are the higher ones, which are all lumped under the term NOx. Let's see, I'm still learning how to run the projector here. I always like to talk about how little molecules like NO are held together. And I've actually been fairly careful to document where I've been getting images from. This is one that is apparently available in the public domain, and I apologize if I have been mistaken. The license kind of said it had been in the public domain. A nitrogen stashes its electrons, outermost electrons, in a 2s orbital and in 3 p orbitals. Same thing for oxygen. And these little up and down arrows, they represent electrons. One of the things I like about this diagram is that they added this little H1 and H2 set. And there's no electrons in them, but this is an intermediate stage, this H1 and H2, to making the diagram for the NO. Essentially what they've done is taken the s orbital, which is spherical, and the pz orbital, which is a dumbbell-shaped object that is aligned along the line that connects the N and the O. And they've combined those to make two new guys, and then they make the H1s on the N and the H1s on the O combined. I fear I'm getting into way too much detail. Essentially, we end up with one electron way up there in what we call a pi antibonding orbital. This destabilizes the interaction between nitrogen and oxygen. If you remove that electron, you get NO+, and NO+ has a stronger bond than plain old NO does. The other guy to be watching, because NO will turn on you if you don't watch it. The other guy to be watching is this pair right here, which ends up being the lone pair of electrons on the nitrogen. So there are some views of what these look like. Here's some little thumbnails that I grabbed from the exhibits that we have next door. That's essentially what the lone pair orbital looks like. There's got a presence on the nitrogen and on the oxygen. This is what the antibonding orbital looks like. And there's the moon. I think I have, not quite there yet. This is basically some more bonding information. Electrons, as indicated by the little blue line in these diagrams, electrons can be given from some non-metal to a metal, like in a direct straight line. This is what we call a sigma bonding. The line of electron density is basically symmetrical around the line that I've drawn. The more interesting thing to me is this pi interaction. Pi simply means that the electron density is divided in half above and below the molecule. So, for example, if chlorine has a lone pair of electrons in the p orbital, it's sitting in that dumbbell-shaped region of space. But the metal also has orbitals that can accept electron density. So this is essentially how a double bond is built. You have a single bond of just straight-on donation, and then this sideways donation. And the sideways donation is tricksy. It can go from a non-metal to a metal, but it can also, if you have something like carbon monoxide or nitric oxide, go from the metal to what we'll call the ligand. And CO has exactly the same number of electrons as NO+, the NO+ cation, as it were. That is Seth. He's named after god of chaos in the desert. You should see the litter box. So here's a plug for the exhibit next door. I've made some models of orbitals. This is the orbital where the lone pairs on nitrogen sit. These little wooden spheres are supposed to be atoms. The regions of electron density have different colors that represent phase. And this is what a pi star orbital looks like. Most pi star orbitals don't really have the moon in the background. That is an added feature of being able to show these in Second Life in 3D. So I mentioned that we should watch out for the those two orbitals, the pi star with that one electron in that lone pair. So those are the orbitals that interact with metals. I love things like nitric oxide, small molecules that interact with metals, because the metals can change the base reactivity of the small molecule in very interesting ways. So this thing with the two lobes and the donut around the middle, I kind of think of it as a p orbital that's gone into middle age, because I've started also to develop a donut around the middle. It can accept electron density into one of its lobes. So in fact, a pair of electrons from the nitric oxide get given to the metal. This is a bonding interaction and serves to keep the NO attached to the metal. Okay, there's also, that's a sigma bonding. So it's basically atom, and the last atom, the metal atom, are all in a straight line. There's also pi bonding. And I've shown pi bonding here. So this big red thing that's the same color as my laser pointer is the metal atom. There is a d orbital that looks like a four-leaf clover, and it's got exactly the right shape. I would love how these things fit together. It's got exactly the right shape to interact with that anti-bonding orbital. So essentially, while the metal steals some electrons from the nitrogen lone pair, it gives them back over on the side. Okay, and that redistribution of electron density allows for the chemistry of the bound NO to be different than the chemistry of regular NO. Okay, so that's a plug for inorganic chemistry. I was going to show you some nitric oxide, but nitric oxide is a colorless gas. Showing it to you, I could have really just gotten away with like an empty flask, but I didn't feel that was right. So I thought I would show you a derivative. So here's nitrosyl chloride. It's not trivial to make, let's say, but this is a photograph from my lab. I made this with students about, when was this photo taken? Oh, about a month ago. And it's called nitrosyl chloride. It's this kind of dark red stuff. Its melting point is minus 59 degrees Celsius. Its boiling point is minus 5.6 degrees Celsius. We essentially generated this by dropping potassium nitrite into hydrochloric acid. And whatever vapors were generated, we directed through a tube that was cooled at liquid nitrogen temperature. Eventually we melted and transferred the material back into this round bottom flask. So that was an afternoon's worth of work. Let's see, why was I doing that? Well, one of the things that I've been doing with my students has been to explore, as part of our senior inorganic teaching lab, some of the chemistry of this material. And it's a molybdenum. It's got two NOs attached. It's got two chlorines attached. That's not enough to satisfy the molybdenum, because molybdenum is greedy. And so basically the molybdenum forms a polymeric structure where chlorides can bridge between molybdenums. And so I've kind of drawn a chain of octahedra that show how the molybdenums can share the Cl atoms to satisfy themselves. To make this, we start with molybdenum hexacarbonyl. It's an octahedral molecule. It's got molybdenum and 6 carbon monoxides. Kind of poisonous, but otherwise well behaved. I'm thinking about some of the relationships I've had. Molybdenum hexacarbonyl is kind of poisonous and well behaved. That fits. We have the nitrosyl chloride. You should call it ClNO, because the Cl is actually attached to the N. And when all the dust settles, we get a beautiful green solid. I'll show it to you. So we made some of this, and it's an air-sensitive solid. It sadly decomposes. It doesn't catch fire. It's not pyrophoric or anything, but it sadly decomposes in the presence of air and moisture. It turns into first a green goo and then a brown goo. So that's why I've showing it to you in the filter that we use. This would be an all-glass apparatus. Everything we do is under a night atmosphere of argon in carefully dried and degassed solvents. So this green stuff was enough for my students to use to play with and use as a starting material to make some interesting compounds. So that's an example of a nitrosyl compound, but there's nitrosyl compounds all around you. Oh yes, there are different ways that NO attaches to metals, and it basically depends on how many electrons are already on the metal. If you look at the d orbitals, many metal atoms already have 6 electrons in their d orbitals. That's nice. What happens is that you get a metal NO with a 180-degree bond angle. It's a nice linear unit. If you get one more electron on the metal, so 7, that 7th electron actually has to go into a higher set of orbitals. And what the whole compound does is reoptimize its structure so as to lower the overall energy of the electrons. The bottom line is that the metal nitrosyl unit can bend. So with 7 total electrons, it bends by 30 degrees. With 2 electrons, it'll bend by 60 degrees. 3 electrons, it won't bend anymore. Those electrons have to go somewhere else. So this affects the chemistry, especially in biological systems. If it's linear, a little bent, or very bent, it's going to have some different chemistry. In fact, the very bent form is related to azanone. Not really going to talk about HNO, but the biological functions of HNO appear to be, let's say, orthogonal or complementary to what NO does. Alrighty. Sounds like a very negative talk. I'm saying NO all the time. So first, let's talk about the atmosphere. So we need nitrogen and oxygen to make NO, but we also need energy. That's nice because it allows our atmosphere to be stable. Oxygen is very reactive, but nitrogen is very unreactive, so we don't have to worry about the entire atmosphere turning into poisonous gas. So we can make NO. We can make NO2 simply by adding enough energy to nitrogen and in the presence of oxygen to burn it. NO is really good in the right amount in the right place. It's really bad in large amounts in the wrong place, even in small amounts in the wrong place. So ground level ozone formation is a significant problem for air pollution. Gives very bad quality of air, and my next slide kind of shows why even a little bit of the NO is a problem. Okay, well, I guess I lied to you. This is how nitrogen oxides can interfere in the ozone layer in the upper atmosphere. So the usual cycle is shown up here in these 3 steps at top. Oxygen in the first step can absorb some UVC light. UVA, UVB, UVC are 3 different wavelength regimes of ultraviolet light, and UVC is, I think, shorter than 250 nanometers. It's really kind of on the border of almost being X-rays. UVC light is really nasty, and it does not penetrate the atmosphere. Oxygen, nitrogen, those molecules will absorb UVC light. But when oxygen absorbs UVC light, it fragments into 2 oxygen atoms. Well, those oxygen atoms in this 3rd step can combine with an oxygen molecule. After the oxygen atoms have bounced around a bit and lost some energy, they can combine with an oxygen molecule to give you ozone, the O3. The O3 will absorb UVB light and some UVC light to give the O2 back again, plus oxygen atoms. And again, the oxygen atoms are going to lose some energy through collisions and recombine with O2 to make ozone again. So these second and third steps end up being a catalytic cycle that converts ultraviolet light into just heat in the atmosphere, which protects us down below from having the ultraviolet light hit us and cause skin cancer and things like that. NOx's can interfere with this because nitric oxide can react with ozone to give you NO2, the nitrogen dioxide, and regular oxygen again. So that screws up the amount of ozone we have. The NO2 can scavenge oxygen atoms and make O2 and make NO again. So those two reactions not only serve to destroy the ozone itself, but destroy the chain carriers, we're going to call them, in the process up here, the second and third step. Because if you get rid of the ozone, this normal cycle can't happen. If you get rid of the reactive atoms, it can't happen either. Now, admittedly, this process is far less efficient. The NOx process is far less efficient at interfering in ozone's job than chlorine atoms are. Chlorine atoms are thousands of times better at screwing up the ozone layer. But this is a thing that can happen in the upper atmosphere. In the lower atmosphere, we can get dinitrogen or nitrogen dioxide from cars, can be hit with light. And in my last talk, I used this little figure to indicate when a molecule was hit by light, gets up into a high energy state. That light can split the nitrogen dioxide into NO and an oxygen atom. Well, the oxygen atom has no trouble finding an oxygen molecule and making ozone right at the ground layer where you can breathe it. Delightful. In a much slower process, the nitric oxide can recombine with oxygen to give us the nitrogen dioxide again. And this nitrogen dioxide can absorb more light. This is a catalytic cycle for making ozone at ground level. So that's a problem. At ground level, ozone has very bad effects on people's health. Hurts lungs. It will oxidize volatile organic compounds, not only from car emissions, but from that are emitted naturally out of trees and the like. And these volatile organic compounds turn into solid particles, aerosols in the air, and again contribute to the formation of smog. I think I've got some pictures of that. Yeah, so this is from satellite data. Let's see. All right, this is from satellite data, and here's a link. This particular map is showing high levels of nitrogen dioxide. The article it came from was very positive that in the United States, the nitrogen dioxide levels have actually decreased because of legislation passed in mid-2000s to control emissions. But you can see that there are plenty of places in the world where nitrogen dioxide and therefore smog is a problem. This is ozone, but of course the ozone is generated from nitrogen oxides. Um, this was recorded from, I think again, 1999. There were extensive fires in Indonesia at the time, um, and their white is aerosol particles, the orange and red is nitrogen dioxide, and well, the orange and red is more of the ozone, which indicates the presence of nitrogen dioxide. Remember, the formation of ozone is going to be driven by light. So during the night, the ozone levels, as it reacts, the ozone won't get replenished. But during the day, the ozone level will spike. There are daily cycles in the ozone levels. So I think another link, I think this is to the same thing, shows up there. So NO has been detected on Mars. This is data from the Curiosity rover. Essentially, samples from the Cumberland Basin. One sample in particular was, the experiment is basically taking some powdered rock and watching, using a mass spectrometer, what molecules come off as you heat it from ambient temperature past 800 degrees Celsius. The scale is, you have to take note of the scale. Oxygen is like the 1-to-1 scale, I guess. The nitrogen looks like a big peak, but it's been expanded by 25 times. What gets me is the interesting shape of some of these features, because we know that nitrogen and oxygen react with each other. Nitric oxide and oxygen react with each other. It's probably shouldn't be surprising to me to see some tailing off like this. This pretty much shouldn't be too surprising when you heat things that contain perchlorates up like this. Perchlorates are strong oxidizing agents. They're probably going to be spitting out the oxygen. And if there's any nitrogen that's organic, it will be transformed into nitrogen oxides. Probably the NO that's released is a indication that it wasn't completely burnt up to make NO2. But it's kind of nice to see NO on other planets. So obviously, since we are communicating by internet, we do have to have a gratuitous cat break. This is Tori, and this was Sin. This was taken about 7 years ago, and Sin is much bigger now. He actually had to go to the vet this week. He's a little bit sick, but I think all is going to be well. So, the other role of nitric oxide that's been very surprising is its role in physiology. As I tell my students when I teach in class, I usually talk about the atmospheric stuff, and then I say, well, if you ever get Aladdin's lamp and you want to do good for the world, just leave nitric oxide alone. Don't wish for it all to go away, because while the smog will go away, so will all the living organisms. Because in 1998, Nobel Prize for Physiology and Medicine was given to Robert F. Fergot, Louis Ignarro, and Farid Murad for the discovery of nitric oxide as a signaling molecule in the cardiovascular system. And in fact, it was 1991, 1992 when these discoveries were made, and in 1992, Science made nitric oxide their molecule of the year. Because honestly, I mentioned EPR detection. These EPR signals had been known for many years in biological systems to signal the presence of some molecule that was having a great physiological effect. It was relaxing smooth muscle tissue, which surrounds arteries and makes blood pressure go down. Everyone thought that it was a huge, delicate molecule that was making the signal. So it was a real revelation when it was a small molecule, NO, a tiny little ubiquitous molecule that's thought to be a poison, is actually a vital signaling agent in the human body and pretty much other, all other animal life. So nitric oxide, it's magic, has a role as a vasodilator. That's the signaling I just mentioned, relaxing smooth muscle tissue to lower blood pressure. It's also a neurotransmitter. It's thought to transmit the pain signals. So it's basically emitted at the end of one nerve, and the neurotransmitter bridges the gap to the next nerve so it can continue sending the signal. And the final thing is that's known about NO in terms of its biological role is in the immune response. Your white blood cells secrete NO because NO is so nasty that if bacteria encounter NO, they tend to die. It's kind of like bleach. So there are a number of biochemical roles in the human body for nitric oxide. So I'm going to show you some structures about, how nitric oxide is managed in the body. So this thing is rat neuronal nitric oxide synthase. It's the whole molecule. And in fact, this top part of the molecule up here is the nitric oxide synthase part. And you may have noticed this large thing that looks like a big blue brain hanging over us in the lecture theater. That's a version of a nitric oxide synthase. And the nitric oxide synthase basically takes a little molecule called L-arginine and splits off a nitrogen from it. There's a heme group in there, I'll mention what that is later, that actually does most of the management. This particular view of the whole nitric oxide synthase has the business end at the front. It has a middle subunit, the green subunit that links the top and bottom enzymes together. The middle unit contains, is basically called comodulin, and it regulates the flow of electrons into the nitric oxide synthase. And at the bottom, there's a reaction that frees up some electrons that can be used eventually by the nitric oxide synthase to make the NO. So let's see. So nitric oxide ends up being made from this molecule called L-arginine. The only thing that changes is this little N group, this guanylyl group right at the top. And essentially, there's a NADPH, that's a energy storing molecule in under physiological conditions. It takes oxygen, and the NO synthase, basically that big blue thing hanging in the sky above us, mediates the removal of an H- from this NADPH, slaps it on one of the oxygen atoms on the O2 to make water, and then there's an OH that ends up being attached to one of the nitrogens on the L-arginine. This NOLA thing is an intermediate, and the nitric oxide synthase basically has to remove the remaining hydrogen and cleave that nitrogen-carbon bond. And it does so to make a final molecule called L-citrulline and the NO. Okay, the business part of the molecule. So here's another view of the same nitric oxide synthase. Down in here we can see a heme group. Here we can see a pterin. It's basically a path to manage the flow of electrons. And this view right here shows a expanded view of the heme group, which actually is the business end. This particular paper I got this structure from has a molecule binding to the heme group to inhibit the nitric oxide synthase. So why would that be important? Okay, yes, so this shows a expansion of the heme group to show you where the iron atom is sitting. And I like these magic eye views. Here's a stereographic view of the same nitric oxide synthase. I'll be setting up this viewer, I guess, in the exhibit in the Science Center Consortium lands after my talk. So you guys will be able to flip through and look at all of these. I didn't have time this time to set up the billboards behind the lecture like I did last time. I think this one is the same one that we had before. So this is a cross-eyed view stereogram. One can basically position the camera at right in the middle at a medium distance, cross your eyes, and eventually you should be able to focus and see this in 3D. Okay, here is a stereographic view of the heme group. I keep using that word, heme group. Basically, the heme group is an iron atom. So here's an iron atom, that kind of browny orange thing, and it's held in place by 4 nitrogen atoms. And those 4 nitrogen atoms are each nitrogen is part of a 5-membered ring. And there are 4 of them, each held together in place by a carbon atom. And there's some other organic shrubbery on the periphery. Essentially, the heme group defines a plane, and the NO attaches on one side of the plane, and in this particular view, we have a cysteine residue. It's got a sulfur atom, and it's binding to the part of the molecule on the other side from where the NO would sit. So in this particular paper, they blocked the active site with this molecule. I'm not even going to attempt to read its name. Reading its name would really just be a stunt. And suffice to say, it's kind of like the L-arginine, which is what the nitric oxide synthase usually acts on, and it can kind of fit into the active site. But once it's there, it just gets stuck. And so it deactivates the active site. And so here is a view on the other side of where we were 2 slides ago. And you can see the 3D view makes it much more clear, but you can see that molecule from the previous slide just sitting like some horrible little goblin above the iron atom and preventing any of its normal substrate, the L-arginine, from getting in there. So this is a potential drug. Essentially, if there is a disease where nitric oxide is overexpressed, that can cause problems in the body. So having inhibitors for nitric oxide synthase to calm down the amount of NO that's being produced is a viable research area. It's also useful sometimes just for pure research to see what effect slowing down the rate of nitric oxide formation has on the signaling process down the line. Okay, so the synthase molecule makes NO, but it's not just enough to make NO. If it just keeps pumping out NO, the NO is going to start reacting with parts of molecules in the body that we don't want it to. NO is quite reactive. Its half-life under these conditions is about 7 seconds. So it doesn't hang around too much before it reacts with water or the like. But when it reacts with water, it's probably going to make some nitrous acid, and that is a whole set of problems in itself. So once you make NO, you got to do something with it. The receptor, the thing that receives the signal, is usually a membrane like this called soluble guanylyl cyclase, or cGMP for short. And that tends to be a fairly small enzyme. What we can see, this is a stereo view again, and you can see, almost clearly see the nitric, or the heme part, where my laser pointer is. I should probably mention that the views that we have here with these red ribbons and spirals, those are called alpha helices, and these yellow ribbons that are flat, those are called beta sheets. There's a lot of atoms that are kind of hidden and not represented explicitly when we show these alpha helices and beta sheets. It's a way of kind of making the molecule seem more simple. Alpha helices tend to have maybe one amino acid that's repeated over and over and over, and that amino acid tends to curl in on itself. The beta sheets, again, have a different amino acids that's repeated over and over and over again, and tends to make a flat sheet. Alpha helices and beta sheets are really common in proteins because they're useful building blocks for making the three-dimensional shape of in this case, the little machine that detects NO. So in fact, the title of the paper I got this from was Femtomolar Sensitivity of a Nitric Oxide Sensor. And so these cGMPs are designed to be able, I shouldn't say designed, have evolved to, oh dear, there's a whole set of comments on the internet coming. They're properties that they will detect a lot or they will detect very sensitively nitric oxide. All right, how do they do that? Okay, well, there's the close-up of the receptor. In this case, we've got an oxygen molecule attached to the iron. It's bent, as you would expect. And, basically, if I had told you this was from heme, it wouldn't be immediately obvious. I mean, hemoglobin, it wouldn't be immediately obvious that I was lying to you. All of these little heme proteins have very similar chemistries. Alrighty. Okay, so we can make NO. We can detect NO. That's our normal function. There's all sorts of little beasties that take advantage of the normal functions. For example, the kissing bug. It has a whole weird little cocktail of enzymes in its saliva, but one variety, or one enzyme is called nitrophorin. And here's a quote: "The saliva of the blood-sucking bug, Rhodnius prolixus, contains at least 7 homologous nitrophorins." And so these are all enzymes that have a heme in them. And they're called NP1 to NP7 in order of their relative abundance in the glands. I'm going to show you NP1. This functionality is so useful for parasites that unrelated bugs have their own nitrophorans as well. Um, the kissing bug is quite annoying because it's a carrier for this Chagas disease. And it's essentially a protozoan that'll get in the bites and infect you because what the kissing bugs do is they feed, they make a wound, and then they essentially poop it right there and then like walk all over it, or you itch and scratch. So the protozoans get into through these bites, and it's very unpleasant. So here's a nitrophorin. You can kind of see a heme group there. The idea is that it injects a small protein into you where the heme has NO attached. And the NO comes off. So NO is a vasodilator. What happens is that your capillaries will relax and the blood will flow better. Essentially, when it's injected, the iron turns into iron 3 and the NO can come off. And that's not the only thing that this molecule can do. Okay, so here's the view of the nitrophorin with the nitrogen and oxygen attached. Notice how it's bent. It can kind of come off fairly easily. After the NO comes off, the molecule binds histidine. And histidine would signal your tissues to swell, which would cut off the blood flow. So it has a anti-swelling side effect, if you will, from the vasodilation. So this molecule, this little molecule is quite clever in that it not only increases the blood flow by making the blood vessels expand a little bit, it also makes sure that swelling doesn't cut off its blood flow. Nasty. Okay, so basically there's the histidine attached to the heme unit. So the story so far, and this is Bert, because this is my oldest kitty. He usually comes out, wants to know what's going on, and then goes back to sleep. The story so far is that biological systems can make NO, can sense NO, and that bugs can take advantage of how we use it. But you always have to remember the nitric oxide is fairly toxic to us. So we have to figure out how biological systems can manage it and get rid of it if it shows up in places where it's not supposed to be. So here's a molecule called nitric oxide reductase, and it actually comes from a bacterium. This is a transmembrane protein. So it actually straddles a cell membrane. A little bit of it is on the outside of the cell, a little bit of it is on the inside of the cell. For these transmembrane proteins, I'm not a biochemist, but I don't even pretend to be a biochemist, well, except on Halloween. When we look at transmembrane proteins, the thing that strikes me is that we often see a lot of alpha helices, and they tend to be parallel or slightly coiled, and this parallel arrangements of the alpha helices tends to be one where the that makes it so that the enzyme can fit into the structure of a cell membrane and be anchored there. It ends up that there's 3 porphyrin units or heme units in the enzyme, and its role is basically to take NO, reacts it with NADPH, you essentially get back to making water and N2O. N2O is a lot less toxic to bacteria. Eventually, the N2O is going to react and make dinitrogen N2. Alrighty, so there's some views. This particular enzyme is quite fun. You know, the inorganic chemist in me loves to look at enzymes that have metals in them. This particular metal or the, enzyme has 3 porphyrin units. One of these porphyrin units has a little friend. It has a little iron atom that's not in a porphyrin attached to it. The yellow, there's also a copper atom, and there's a calcium atom circled in blue. I think the calcium atom is mainly structural. It kind of is there to pull the helices to make sure they're in the right place and kind of tightly bound to each other. This copper atom, I don't know what its function is. That's still under some debate. So what is this friend? Why do atoms have friends? Do atoms have friends? So here's the heme unit. I've given you a one of those cross-eyed stereo views, and essentially there's an iron, an O, and an iron, and those 3 atoms are pretty much in a straight line. Okay, so we've got what I would call a mu-oxo dimer. Mu means bridging. Oxo is what an oxygen atom is called when it's bridging, and it's sandwiched in between the 2 iron atoms. We've got an example here of a heme attached to a non-heme iron. I haven't talked very much about non-heme irons, and I don't want to give you the wrong impression. This iron atom is actually attached to various things various atoms in the protein. It's got probably 6 other bonds to it, including the oxygen that's also attached to the other iron atom. This it's very likely that this bridging iron atom is one that is very important to the function of the NO reductase. It probably gets attached to the nitric oxide at some point. Okay, so I'm getting close to the end of my talk. So what was with that first slide? Okay, I showed you meat, and I showed you Viagra and stuff. Well, here's the thing. That nice red color of the meat that makes it look fresh, well, that's actually due to a myoglobin NO complex. And those are usually formed when nitrites that might be in the plastic wrap are touching the meat surface. So the nitrites get reduced by the myoglobin in the meat, and that forms NO. And it turns out the myoglobin NO species is bright red. Otherwise, the meat would be kind of unappetizing and grayish. This is the same sort of thing that happens in cured meats. So cured meats end up having some nice red colors to them. You would use salts to cure meats. Most salts, curing salts, have a tiny touch of sodium nitrite, which is NO2 minus, which reacts with the proteins and gives you a little NO. I also showed you the Viagra and Cialis for the erectile dysfunction drugs. Well, NO regulates blood pressure by vasodilation. When I idly Googled nitric oxide, I got a lot of hits for nutritional supplements like L-arginine, and the rationales were things like, well, as you age, nitric oxide levels in the body go down, so you need these supplements. The sad thing is that just having extra L-arginine in your diet is not going to automatically make more nitric oxide. The nitric oxide is made by the enzyme, and if your enzyme levels have gone down, there's no way of getting those back up just through diet. If the enzyme levels have gone down, then you won't be making as much nitric oxide, no matter how much of the starting material you have. I kind of think of it as, I don't know if you remember, there is a episode of Lucille Ball, where Lucy and Ethel are in the chocolate factory, and they're putting chocolates in boxes, and the chocolates start coming out faster and faster and faster. Well, Lucy and Ethel are the enzyme. The chocolates are the L-arginine. It won't matter how fast the L-arginine is coming out. The boxes won't get filled any faster. So the problem is, if there's less NO being produced, maybe if we make it so the NO doesn't get destroyed as fast, the lifetime of each NO is a bit longer, and each NO does more signaling. That's what an NO reuptake inhibitor does. Basically, drugs like Viagra and Cialis are ones that block the sites that destroy the NO. It means the NO lasts a little longer, does a little more signaling, a little more blood flow happens. Yes, indeed, early ED drugs had to be injected. And let me get you that particular reference. It's worth a quick look. So in fact, Dr. Brindley injected it at a conference and then offered to do a live demonstration. During the conference, when he was giving his talk, he dropped his pants and allowed everyone to inspect the results of those injections. I think he'd be arrested today for doing that. What we do in the name of science. So nitric oxide. Nitric oxide does a lot of things for us. It allows us to live, the way we do. It is a fundamental small molecule. It was the first tiny molecule that was discovered to have a physiological role as significant as it does. People are now thinking little molecules like HNO and carbon monoxide, CO, also have such important roles too. All right, closing in on the end of my talk. I'm like to acknowledge my students, as always. Since 1998, I've had like 30 master's students and maybe 20 master's students and 30, 35 undergraduates working with me. We don't have a PhD program here. My collaborator, George, at the University of Oklahoma, I tried to send students down to him to get their PhDs, working in this area of nitric oxide, heme type chemistry. As always, my department, College of Arts and Sciences, the graduate school here at SIU, have been very supportive. Currently, NSF award from 2012. We have been renewed, so we'll be continuing to work in the next 3 years or so on the HNO molecule and how it interacts with transition metal compounds. Let's see, finally, where I get stuff from, I try to do everything public domain, try to make sure that all of my sources are cited. We have cats up here. There's Sin and Tori and Seth. And you'll notice that Tori doesn't look too much different from my sketch of him. There's a blob sitting there with orange colors on him. So, where are we? I think that's it. So that is my talk for today. I'm happy to take questions. I'm less happy to take abuse, but I'll take it. Basically, I'm going to be here for a few more minutes. So Let's see. Are those his active bonding sites? Yes. My cats actively bond. Um, when they're asleep, at least. Thank you. Alrighty. Well, I'm going to do a quick pan. No does mean no. Quick pan, and we see the audience and the anno floating in the sky, or the synthase floating in the sky. It reminds me of that an episode of Futurama. My favorite quote from Futurama. Question: what caused the extinction of the dinosaur? Answer: me. Why not Zoidberg? Well, in the new lab, we just got a new building about 3 years ago. In the new lab, I do not have to walk sideways. The old lab, the benches were a little bit close together, and if there were a couple of people in there, you kind of had to scuttle from side to side. All right. Well, with that, I am going to stop.